In our last segment for Module 2, we'll examine the structures that are inside of the cell. The cytoplasm is going to represent all of the cellular materials and substances that are located between the plasma membrane and the nucleus of the cell. These materials consist of the cytosol, the organelles, the cytoskeleton, and any inclusions. So recall that the cytosol typically refers to the gel-like fluid within the cell. Inclusions include things like pigments such as melanin, which gives our skin and our hair its pigment or its color, fat droplets, or even glycogen granules. Glycogen we see is a starch that is produced by animals, and it serves to store glucose until our body needs it. So here we'll briefly review the organelles as well as the cytoskeleton. So the nucleus is the largest and the most prominent organelle of the cell. The nucleus is so large that it is very easy to see as a dark oval that's usually near the center of most cells. It functions to control most of the cellular processes, and it also contains the DNA of the cell. The DNA is the cellular instructions for making every protein within that particular cell. So most cells in the body have a single nucleus. Some cells, such as the skeletal muscle cells that are seen here, have many nuclei, and we call these types of cells multinucleate. Some cells, like mature red blood cells, which are called erythrocytes, have no nucleus, and we refer to this condition as anucleate. The mitochondrion is a capsule-shaped organelle that is usually found scattered throughout the cytosol. This organelle contains two membranes. It has an inner membrane that has many specialized enzymes on its surface that catalyze the formation of ATP. And ATP, recall, is abbreviation for adenosine triphosphate, and it is the cellular energy molecule that the cell uses to fuel most of its processes. We see that the mitochondrion will produce most of the ATP for the cell. Some of that ATP is produced in the cytosol, but not all of it. The majority of it is produced by the mitochondrion. And typically we see that cells that produce lots of ATP, such as skeletal muscles or kidney cells, tend to have a lot of mitochondria, so physiologically that makes a lot of sense. The endoplasmic reticulum is an organelle that is typically continuous with the plasma membrane of the nucleus, which is called the nuclear membrane. The endoplasmic reticulum generally appears as a series of flattened channels that are interconnected to one another, and these channels are called cisternae. The endoplasmic reticulum comes in two varieties or two forms, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So let's examine these in order here, starting with the rough endoplasmic reticulum. So the rough endoplasmic reticulum is called rough because its surface is studded with small structures that are called ribosomes. The ribosomes function in protein synthesis, while the tubules of the rough endoplasmic reticulum function to store the proteins that are made by those ribosomes, as well as transporting proteins to other parts of the cell. So ribosomes are an organelle that lack a membrane. They can come in two forms. They can be bound ribosomes, which are associated with the rough endoplasmic reticulum, or they can be free ribosomes that float around within the cytosol. The ribosomes are the sites where proteins are synthesized from messenger RNA molecules. That is, the messenger RNA tells the ribosomes which proteins to manufacture at that point. And then the ribosome actually has the job of linking the amino acids in that particular protein together in a chain. So next we have the smooth endoplasmic reticulum, which has no role in the synthesis of proteins, but it does function to produce things like lipids, phospholipids, as well as steroid hormones, which are, as we've discussed in previous segments, are lipid-based because they're based off of the cholesterol molecule. The smooth endoplasmic reticulum also functions to detoxify alcohols as well as drugs. So for these reasons, smooth endoplasmic reticulum tends to be really abundant in certain endocrine glands, such as the ovaries and the testes, where they produce steroid sex hormones. And in addition, they're also abundant in the liver because we see that the liver functions to cleanse the blood of things like alcohols and drugs that have been ingested. Next, we have the Golgi apparatus, and the Golgi apparatus kind of superficially resembles the interconnected flattened channels of the rough endoplasmic reticulum, except we see that the Golgi apparatus has no ribosomes on its surface. The Golgi apparatus functions in modifying the proteins that are made by the rough endoplasmic reticulum as well as the ribosomes. And then what it does is it packages those proteins into small little bubbles called vesicles, and those vesicles will distribute the proteins to different parts of the cell, or they will transport them for export from the cell. The modified proteins that are placed in these vesicles, the vesicles as I've kind of described, are actually small spherical bubbles that are constructed from phospholipids. So they're made of the same stuff that the plasma membrane of the cell is made from, and the same stuff that all the membranes of most of the organelles that we've seen are also constructed from. So given that we've talked a lot about protein synthesis and protein distribution around the cell, let's take a moment and put all these different things together. So we've learned that the DNA inside of the nucleus contains the instructions for making proteins. However, for the most part, DNA is trapped within the nucleus. That is, it cannot leave the nucleus. And we also just saw that all of the protein-making machinery, so the ribosomes, the rough endoplasmic reticulum, and the Golgi apparatus are all located outside of the nucleus. And so what we see that happens is that the DNA is translated into another molecule called RNA, and a certain type of RNA that's made from the DNA template is called messenger RNA. And messenger RNA has the ability to leave the nucleus and work with the ribosomes, where that RNA is then translated into proteins. And the ribosomes will read the RNA, and then from reading the RNA, they will link the amino acids of that particular protein together into what will become that protein. Then finally, we see that the Golgi apparatus will put any finishing touches on these proteins, and it's going to package them into those plasma membrane vesicles for transport from the cell, or transport to different parts of the cell. Next, we have lysosomes and peroxisomes. These are small, kind of sac-like organelles, so they're going to look, at least in diagrams, very similar to one another. Lysosomes are small sac-like structures that contain digestive enzymes, and lysosomes function as demolition organelles. So the enzymes that are contained within them are used to break down damaged organelles, worn out cell parts, or they're also used to destroy pathogens like bacteria. So we'll see that lysosomes not only function in the breakdown and destruction of pathogens like bacteria or fungi or things like that, but they also function in cell destruction. So if cells are very badly damaged or if they're unhealthy, they can initiate a process called apoptosis, which is programmed cell death. The lysosomes of that cell will then rupture when apoptosis is initiated, and it will release those digestive enzymes into the cell, which will then kill the cell that has released the enzymes from those lysosomes. Peroxisomes, even though they look very superficially very similar to lysosomes, are going to have quite a different function from them, but they do have a similar function to the smooth endoplasmic reticulum, in that peroxisomes help to detoxify things like alcohols and drugs. The enzymes that the peroxisomes contain are called oxidases because they use oxygen as part of their enzyme structure to detoxify drugs and alcohols. And in doing so, they create hydrogen peroxide in the process. These organelles are particularly abundant in things like the liver, which, as mentioned previously, serves the role of detoxification of the blood from things like alcohols and drugs. The centrioles are an organelle that appear as these paired cylindrical bodies that tend to lie in fairly close proximity to the nucleus. They function as the origin point for all of the microtubule cytoskeleton elements, which we'll see here in a moment. They also function in moving DNA strands during cell division, which is also called mitosis. So now let's examine the cytoskeleton. So now that we've described the functions of the organelles, we'll turn our attention to the cytoskeleton of the cell. The cytoskeleton is a series of protein filaments and tubules that internally support each cell, and they give the cell its structural integrity. So as the name implies, they provide roughly the same function that our bony skeleton provides our entire body, except they're made out of protein instead of calcium. The protein filaments that make up the cytoskeleton are also important not only in cell structure, but also in the functions of cell motility, which means cell movement. They're also important in mitosis, as well as transport of substances within that particular cell. So there are three elements of the cytoskeleton: the microfilaments, the intermediate filaments, and the microtubules. So the microfilaments are the smallest in diameter of the cytoskeleton elements, which is why they're named as such. They are essentially chain-like threads or filaments that are composed of individual proteins called actin. Microfilaments are important in that they function in cell motility, and we also see that they're very abundant in muscle cells, particularly things like skeletal muscle and cardiac muscle, which are contractile tissues. The intermediate filaments, on the other hand, as their name implies, are intermediate in diameter to the microfilaments and the microtubules. They are formed from protein filaments that are called keratin, and these keratin filaments are wound around one another like the individual filaments of a rope. The intermediate filaments, much like a rope does, functions to resist tensile forces, so that is, they resist pulling and stretching forces that are applied to the cells. So for example, the skin has abundant amounts of intermediate filaments, particularly within the cells of the epidermis, that make it very resistant to stretching and pulling forces that are applied to it. Inside of the cell, the intermediate filaments also help to maintain the cellular shape and structure, but they also serve as an anchoring point for organelles, so they function as kind of an internal scaffolding. The intermediate filaments that are shown here in purple are the filaments that provide the internal support for desmosomes, which we saw are cell-cell junctions that link nearby cells together, and they help link those cells together in a continuous sheet, so that way that sheet of cells in that particular tissue can resist tearing. Finally, we have the microtubules, which are the largest in diameter of the cytoskeleton elements. They are tube-like structures that are composed of many bead-like proteins that are called tubulin. So unlike the intermediate filaments, their function is to resist compressive forces that are placed on cells. And they also act in kind of much the same way that the intermediate filaments act. They act as an internal scaffolding, except this time the microtubules help to resist compressive or crushing forces, whereas the intermediate filaments help to resist those tearing or tensile forces. We'll see that these are also the structures that give support to the cilia and flagella, which are important cellular extensions that exist in certain groups and populations of cells throughout the body.